Hi friends, welcome back to Anand Science Academy. In this video, let's discuss an interesting topic that is codominance. Till now, we were discussing crosses where the F1 resembled either of the two parents in the case of dominance pattern of inheritance or was in between, that is in case of incomplete dominance. But in the case of codominance, the F1 generation resembles both parents. A good example of codominance is different types of red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by the gene I. The plasma membrane of the red blood cells has sugar polymers. These sugar polymers we call as antigens. They protrude from the surface of the RBC and the kind of sugar is controlled by the gene I. The gene I has three alleles IA, IB and small I. The alleles IA and IB produce a slightly different form of the sugar, while allele small I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three I gene alleles. IA and IB are completely dominant over small I. In other words, when IA and small I are present, only IA expresses because small I does not produce any sugar. And when IB and small I are present, IB expresses. That's why we call as IA and IB are dominant alleles and small I is the recessive allele. But when IA and IB are present together, they both express their own types of sugars. This is because of codominance. Hence, red blood cells have both A and B types of sugars. Since there are three different alleles, there are 6 different combinations of these three alleles that are possible. And therefore, a total of 6 different genotypes of the human ABO blood types. Here on the left side, we can see the 6 different types of genotypes. IA, IAIB, IA small I, IB, IB small I, small I. The right side, the tabular column is given. It is also given in our NCERT reader. There are 7 types given. Don't get confused with that. IA, IB are given 2 times. So, 3 different types of alleles in combination produce 6 different types of genotypes. Be clear with this. With these 6 genotypes, how many phenotypes are possible? Let's see. There are 4 different phenotypes are possible with these 6 different types of genotypes. They are IAIA, A group, IAIB, AB group, IA small I, A group, IB, B group, IB small I, B group, small I, O group. Hope you are able to follow, my dear friends. Do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than 2 alleles, in this case 3 alleles, governing the same character. Since in an individual only 2 alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has 2 alleles, capital B and small b. Starch is synthesized effectively by capital B homozygotes, and therefore large starch grains are produced. In contrast, small b homozygotes have lesser efficiency in starch synthesis and produce smaller starch grains. After maturation of the seeds, capital B seeds are round in shape and the small b seeds are wrinkled in shape. Heterozygotes, that is capital B small b, produce round seeds, and so capital B seems to be the dominant allele. But the starch grain produced are of intermediate size in capital B small b seeds. So, if starch grain size is considered as the phenotype, then from this angle, the alleles show incomplete dominance. Hope you are able to follow, my dear friends. In this tabular column, the genotypes and the different phenotypic expressions are shown here. The genotype capital B produces large sized starch grains and round shaped seeds. Capital B small b genotype produce intermediate size starch grains with round seeds. Small b genotype produce small sized starch grains and wrinkled seeds. Here, with respect to starch grain size character, the gene shows incomplete dominant pattern of inheritance. And with respect to seed shape, the same gene shows dominant pattern of inheritance. Therefore, dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine. In case more than one phenotype is influenced by the same gene. Okay friends, I hope you have understood what is codominance and multiple allelism and how multiple alleles govern a single character and also an example for a single gene controlling more than one character. Let's continue the chapter in the next video.